This presentation is a case study about Pyruvate Dehydrogenase Deficiency. PDC is an X-linked recessive disorder that affects many genes, but the most common of them is the gene PDHA1. This gene is responsible for transcribing and creating the E1 enzyme. Because of this gene becoming mutated, it is unable to produce the E1 gene, which does not allow pyruvate to be broken down, hindering the respiration process of cells. This causes a buildup of lactic acid in the body, which then leads to nausea, vomiting, severe breathing problems, abnormal heartbeat, and neurological problems. Because of the severe health effects, many individuals do not survive past childhood, although some may live in adolescence or adulthood. Teresa and her husband Eric are a young married couple that are expecting their first child. The first trimester passes by smoothly until the 22nd week mark, when it is time for Teresa's first ultrasound. During this time, the doctor notices very concerning brain abnormalities and expresses his concern to Eric and Teresa. He assures it is too early to tell if these abnormalities are permanent and is unsure of what they might mean. But until he is able to consult with other professionals and find out what these abnormalities may mean, he encourages Teresa to stay calm and be especially careful during these next few weeks. Teresa becomes stressed, like any new parent would, and begins to research the information that the doctor provided and the symptoms present. She stumbles upon a website that seems to have the same information she heard about. She already knows that she is having a girl by the ultrasound at the 20-week mark. Given the information provided by the doctor at the last appointment, Teresa sees that Pyruvate Dehydrogenase Deficiency is common in girls but abnormal to have. Online, it says that females with Pyruvate Dehydrogenase Deficiency caused by the mutation of the PDHA1 gene often have skewed X inactivation, which results in the inactivation of the X chromosome with the normal copy of the PDHA1 gene in most cells of the body. The PDHA1 gene provides instructions for making a protein called E1 alpha, which is a part of a larger protein. These two E1 alpha proteins combine with two copies of another protein called E1 beta, to form the E1 enzyme. This enzyme is known as pyruvate dehydrogenase and is a component of a group of proteins called pyruvate dehydrogenase complex. She keeps this information in her head before her next doctor's appointment to ask a few more questions and follow up on her research.
The next couple of weeks, Teresa goes to her scheduled checkups and the doctor explains that he would like to do more blood tests and an analysis of her urine. Once the doctor does the tests and the results are in, there seems to be low levels of estriol in Teresa's urine. She is now almost 9 months pregnant, and the doctor decides to wait in order to prescribe anything until after the baby is born. Teresa gives birth to a low-weight baby girl with an abnormally shrunken head with multiple indents and malformations. Once born, the doctor on staff decide the best thing for Teresa's baby is urgent care, and so the baby spends the next few days in the ICU while doctors run blood tests and an MRI scan to determine what could be happening. A couple of days after, the results are in. The blood tests show that the baby has high levels of lactate in the bloodstream, lactic acidosis, and has symptoms of low muscle tone and seizures. The disease was officially diagnosed through checking the pyruvate dehydrogenase enzyme. The doctor concludes from the multiple tests that the baby has a rare form of pyruvate dehydrogenase deficiency and can be released from ICU on instruction. The baby's test results show that the brain of the baby seems to be progressive. However, the head size is very small and has abnormal structures. This is common in babies with pyruvate dehydrogenase deficiency.
Cram.com. Welcome to another MedCram.com Board Vitals question. This one's from USMLE Step 1 and Comlex Level 1 Bank. A 35-year-old female was being evaluated for diabetes. She visited a diagnostic center to get a glucose tolerance test done. The attending staff at the center accidentally administered 10 times the dose of oral glucose which was supposed to be given. After some time, she showed neurological disturbances, and they sent her to the ED by ambulance. At the ED, she was found to have lactic acidosis. The physicians strongly suspect an inborn defect of carbohydrate metabolism. Which of the following enzymes is probably deficient in the patient, which led to lactic acidosis? And your 5 choices are A, glycogen synthase, B, liver phosphorylase, C, pyruvate dehydrogenase, D, phosphofructokinase 1, and E, muscle phosphorylase. Okay, so the thing that you've got to realize is what is the metabolism of glucose? So glucose is taken into the body and it undergoes something called glycolysis. And that's a multi-step process that we don't want to necessarily go through completely. But the byproduct of this is something called pyruvate. And as a result of this, going from glucose to pyruvate, you actually get some ATP. Now, ATP is kind of the currency of energy in the body. So ATP is energy. Without this energy, your body will not work. However, this is not the majority way of getting energy from the body. What the body does is it takes pyruvate and goes through an enzyme called pyruvate dehydrogenase, which I'll abbreviate PD. And that leads us to an intermediate called acetyl-CoA. And acetyl-CoA then goes into Krebs cycle, which goes around and around and around. And the result of that is, in fact, ATP via mitochondria. So this involves NADH, FADH2, and we don't want to get into that specific. So this is kind of the overall thing. Glucose will go to pyruvate via something called glycolysis, and then pyruvate is converted into acetyl-CoA by pyruvate dehydrogenase, and then acetyl-CoA then goes into Krebs cycle, which is right here. It's complicated, and all the way around the cycle over and over again, and you make NADH and FADH2 and even a GTP. It all ends up being ATP after you go through the electron transport chain, which is in the matrix and intermembrane space of the mitochondria. That makes ATP. Okay, so this is the major way that ATP is made. Now, if there is a problem, for instance, right here with pyruvate dehydrogenase, in other words, if there is a shutdown of this enzyme, then what will happen is there'll have to be a switch somewhere. So pyruvate will then have to go somewhere else and be metabolized elsewhere. Now, the thing I haven't shown you here is that glycolysis also takes something called NAD+, which is very important, NAD+, and converts NAD+ into NADH. Okay? So that's a reducing agent, and it's this NADH can actually also go to the electron transport chain and create ATP. Here's the problem. If you have glucose, which is going to pyruvate, and this is the only thing that's occurring, eventually what could happen is you could use up all of your NAD+. Okay? So what the body does is if you're unable to replenish your NAD+ here in the electron transport chain, pyruvate will then be converted and shuttled over to a different pathway. So in other words, if this pyruvate dehydrogenase is shutting down, pyruvate has to be metabolized in a way that will regenerate NAD+ so that you can continue with glycolysis and make ATP in this way. Because if this is shut down, you can't make it this way. So again, if pyruvate dehydrogenase is not working, you're not going to get ATP here. You're going to have to get ATP from here, and to be able to do that, you have to make sure you have enough NAD+. To do that, the NADH has to be converted back into NAD+, and the way to do that is to convert pyruvate into lactic acid. And so therefore, when you see a problem with pyruvate dehydrogenase, you see a shuttling of pyruvate. Instead of going to acetyl-CoA, you see it going to this thing called lactic acid. So that's key to understand. So if we go back to the question stem, you'll notice that our patient here in the emergency room had a large amount of lactic acidosis. And that was after the patient was accidentally administered 10 times the dose of oral glucose, which was supposed to be given. So basically what we're seeing is that in this situation, there's so much glucose that's being made that it's flooding this pathway. And if this is the rate limiting step, or if there's some sort of problem here at pyruvate dehydrogenase, you're going to get an increase in this pyruvate, and the pyruvate is going to be shuttled over to this lactic acid. And the reason is because you have to regenerate this NAD+ so you can get ATP. So based on these answer choices, what do you think the right answer is? And of course the answer is pyruvate dehydrogenase. The problem with it being glycogen synthase is because in glycogen synthase you actually have a problem making glycogen, and you have problems with hypoglycemia, and you have to metabolize in a different way, so you get ketoacidosis, etc. The problem with liver phosphorylase is that you get only mild hypoglycemia, and you get actually infiltration of the liver with glycogen. So that can't be it. We've already said that C is the right choice. Let's look at D. With phosphofructokinase 1, the thing that you've got to remember is that the issue is further up here in glycolysis, because remember, phosphofructokinase 1 is the rate-limiting step for the pathway of glycolysis. So what you actually see is a buildup of glycogen. You see actually very low levels of pyruvate and therefore lactic acid with exercise, and actually exercise tolerance is impaired. Finally, muscle phosphorylase is also known as Mcardle Syndrome, and it looks very similar to phosphofructokinase 1. There's poor exercise tolerance and issues with levels of glycogen, glucose, etc. And so not consistent with lactic acidosis, which is actually low in Mcardle syndrome. So again, knowledge of the pathway, specifically that pyruvate has two fates. One is lactic acid that regenerates the NAD+, and the other is acetyl-CoA, which is the main way that it's metabolized in the Krebs cycle, which forms ATP via the electron transport chain. Thanks for joining us.
What gene is affected? Teresa's doctor sends her home with some information about pyruvate dehydrogenase complex deficiency, and here's how the genes were affected. The PDHA1 gene is one of the most commonly affected genes. Its normal function is providing instructions for making a protein called E1 alpha. The E1 alpha, E2, and E3 proteins are affected. Their normal function is to perform the chemical reaction converting pyruvate to acetyl coenzyme A. This chemical reaction is essential to begin the series of chemical reactions to produce energy needed for cells. In females who have two copies of the X chromosome, one of the altered copies of the PDHA1 gene in each cell can lead to less intense issues of the condition and can cause no signs or symptoms ever. The protein is included to make sure the gene is properly functioning. Proteins control the activity of the complex in which pyruvate dehydrogenase phosphate activates the complex, while pyruvate dehydrogenase kinase inhibits the complex. What is the inheritance pattern? The next day, Teresa calls the doctor asking how the baby could have inherited this, because she doesn't recall anyone in her family having this disorder. He tells her that people who have a pathogenic variant in PDHA1 are the first people in the family with a genetic change, and Teresa probably has some symptoms of any of PDC. This means that each future child that Teresa will have include a 50% chance to inherit the pathogenic variant that causes this. This deficiency is not revolved around sex chromosomes, but autosomal genes. People with changes in both copies will have some type of symptom of the disease. In regards, Teresa may not have any symptoms at all, resulting in being a carrier of autosomal recessive inheritance.
This short video will help break down the basic steps of X inactivation to help you better understand how autosomal recessive inheritance works for this particular disease. It is a very short video, but very important in understanding how this genetic disorder is passed down.
Inactivation is a process that mammals use to equalize the gene dosage between males and females. Since normal male mammals have only one X chromosome and females have two, there is a potential imbalance in the expression of genes on the X chromosome between males and females. In female mammals, this imbalance is prevented by inactivating all but one X chromosome in all somatic cells. X inactivation can be divided into three phases: initiation, spreading, and maintenance. Initiation occurs during embryonic development. Each X chromosome has an X inactivation center, or XIC. The number of XICs are counted, and one X chromosome is randomly chosen to remain active while the other is targeted for inactivation. Spreading is the process of compacting and inactivating the X chromosome. A gene designated XIST within the X inactivation center encodes an RNA that coats the X chromosome and promotes compaction, beginning at the XIC and progressing toward both ends until the entire X chromosome is inactivated. When spreading is finished, the compacted chromosome is called a bar body. After the bar body is formed, maintenance begins and continues through adult life. The bar body is replicated just like other chromosomes, but both copies of the replicated bar body remain highly compacted and essentially inactive. Thus, once an embryonic cell undergoes X inactivation, all cells that derive from it will have the same X chromosome inactivated.
Here we can see how this disorder is inherited from person to person. It can be inherited through one of two ways, either X-linked recessive or autosomal recessive. Because this is a predominantly X-linked recessive gene, the disease is present in males, but because they only have one X chromosome, it is not inherited through them. Rather, it is the mothers who become the carriers. We see this in the autosomal recessive inheritance pattern occurring when female carriers only have one mutated X chromosome but still display symptoms due to the X inactivation, which in turn can be passed on to their child. The next day, the doctor sits Teresa down to talk about treatment options. He recommends a diet that is high in fats and low in carbohydrates. He explains this will assist in reducing lactic acidosis. He also recommends medications to prevent seizures. Teresa leaves, discouraged and upset on what's to come. Because PDD is such a rare disorder, there is still very little treatment for people with this disease. And sadly, despite our knowledge, there's a very high mortality rate.
Teresa begins to notice her baby often has rapid eye movements and rapid breathing. She also struggles to eat. One day the baby has a seizure. Teresa rushes her to the emergency room. Despite the options the doctors give Teresa, they make it very clear that Teresa's daughter will likely not live past her first year, and she should be aware of the symptoms that her baby is experiencing. Despite medication and treatment, Teresa's baby passed away during the first year of life. The provided link leads to a quiz hosted on Kahoot. Take the quiz to test your knowledge on pyruvate dehydrogenase deficiency. The information and resources found in this presentation were gathered from the SSRM College, Cambridge University Press, the National Institute of Health, Rare Diseases and Genetics Home Reference Databases, the United Mitochondrial Disease Foundation, Wikipedia, and YouTube.